clearing the malignancy.
This video is going to be about exosomes and how they can actually work with immune cells. Cancer cells, they can hijack exosomes, but immune cells also rely on exosomes as well. So they are formed in
Exosomes are tiny vesicles released by cells. They carry RNA molecules, including those that bind to CD70-bound RNAs. But pecans contain compounds that inhibit the PI3K-mTOR pathway. This reduces exosome production and release.
It's no wonder that they can help immune cells and also cancer cells. And since their functions are protective, which is the exosomes, it's no wonder that cancer cells hijack them so that the exosomes protect. Exosomes are actually internalized into early endosomes via ESCRT machinery, TSG101, ALIX, and then tetraspanins, CD9, CD63, CD81. I made a separate video about that, and certain foods that actually targets those machinery types in the context or in the attempt to go against cancer, but it might also affect immune cells as well, since immune cells use exosomes too. Exosomes behave like long-range signaling extensions of the parent cell, modes of communication, ligand-receptor binding without uptake, endocytosis, signaling from endosomes, and membrane fusion, leading to cargo release, because endosomes, they do have a lot of forms of cargo. Also, endosomes remove unwanted or excess proteins, RNAs. Okay, so exosomes, they actually pass around unwanted cellular waste export systems, or they function as cellular waste export systems. Example, excess signaling receptors, misfolded proteins, redundant miRNAs or mRNAs. So it sounds like exosomes, they might be able to help the immune system in the sense where the scenario where tumors they soak up the RNAs, or they seem to send out like miRNAs or mRNAs as sponges as an attempt to stop immune cells trimming the poly-A tail on CD70. So the poly-A tail is basically the linchpin of the CD70. That's also what tumors fear. That's well, at least when it comes to CD70, the poly-A tail is the tumor's one of the tumor's weak points. So by trimming the poly-A tail, that's how CD70 is stopped. But that's going to be for another video. Okay, so immune cells use exosomes as information carriers, such as carrying MHC1 and MHC2. Now, MHC1 and MHC2
semi-mature antigen-presenting cells don't activate T cells correctly, leaving T cells untrained.
Because T cells can be naive, and they can't fight properly in battle against cancer cells unless if antigen-presenting cells train them properly. Now, the first food here that could support the immune system cells when it comes to exosomes is red cabbage. So red cabbage has anthocyanins, also cyanidin, 3-glucoside, and immune cargo favors facile CD70 tolerance-biased exosomes.
Fossil, the death receptor system. So tumors evade immune attack by blocking this pathway. To trigger apoptosis, phosphosol must disengage.
By a facile on CD70.
Cold is my weapon, and so is nature's power. Apogene disrupts tumor evasion, restoring immunity.
Sounds good for immune cells. Next is astaxanthin,
found in salmon, protects immune from oxidative damage. During inflammation, immune cells are under attack, but astaxanthin strengthens their defense. By consuming salmon rich in astaxanthin, immune can enhance relieving stress. Nature's pollen, natural folds and antash shield. Astaxanthin, defending immunity.
CD70 on the cancer tumor, then that will lead to ongoing NFKB, leading to exhausted T cells. So it sounds like astaxanthin protects T cells against exhaustion. Targets the machinery of exosomes, upregulating NS MASE2, so NS-MACE, that's one of the machinery for exosomes. So by upregulating that, NS-MACE2, that increases exosomes. Net effect, high output, anti-inflammatory exosome release. Okay, next one here is a grapeseed extract. Upregulates NFKB P50. Upregulates NR4A, which could be good when it comes to sustaining AP1. Also, grapeseed extract limits surface display of CD70. So that makes grapeseed extract really good, because that means that CD70 is not shown on the surface of a tumor. CD70 might even be internalized. Therefore, CD70 can't suppress CD27 on an immune cell, because if it does, then that's ongoing NFKB and then eventually AP1 loss, leading to exhausted T cells. Okay, so signal dense but low inflammatory exosomes as a result of grapeseed extract. Okay, flaxseed meal has ALA, a form of omega-3, also lignans. So flaxseed meal lowers NFKB, P65 transactivation. All right, so sounds good against CD70. Also, flaxseed meal lipid remodeling, upregulating
power. Tumors use exosomes in the tumor microenvironment. Too many mutant Tregs suppress immune response. Time to clean up.
Via PUFA ceramide coupling. So,
but some
like
threats need a stronger hand. And now, the final solution.
For
Watermelon.
So NS-MACE2 is the machinery, is one of the key machinery for exosomes. So therefore, supporting immune cells. The next is a watermelon, has a citrulline, increases HIF-1 alpha via nitric oxide. Of course, because arginine creates a nitric oxide, upregulates vesicle release pathways, neutral on cargo loadings. Okay, so exosomes, they have different types of cargo with different purposes for each
watermelon,
cargo.
rich in citrulline, which your body converts to arginine. Arginine boosts nitric oxide.
Therefore,
nitric oxide disrupts tumor metabolism, inducing oxidative stress and apoptosis. Fuel the fight from watermelon to warrior.
Watermelon, it does have a citrulline. It helps in ARG1, arginine depletion in the hostile tumor microenvironment by tumors. So tumors hog up the arginine and then starving T cells, because T cells need arginine. So watermelon and citrulline, it selectively replenishes arginine for T cells, but not for tumor cells. Okay, so this could be how cancer cells, they actually win and they thrive and survive. Like let's say, for example, here, tomato lycopene, immune cargo on exosomes, upregulation of CD70, upregulation of PDL1 co-packaging. Now, those are CD70 and PDL1, those are very suppressive and antagonistic against immune cells. So, eating tomatoes could possibly be supporting that, unfortunately. But tomatoes have other very important anti-cancer benefits as well. Okay, the way exosomes initiate CD27, CD70 axis is step 1, CD70 packaging. So tomatoes, they unfortunately upregulate the packaging of the CD70. And then there's the CD70 promoter, NFKB responsive, AP1 independent. Surface 70, CD70 is downregulated under chronic signaling. Exosomal CD70 is preserved and enriched. And then target cell engagement, receptors, memory T cells, Tregs, and exhausted CD8 T cells, they all have CD27 receptors. And then adhesion molecules, ICAM, on the exosome. So that's it for the video. Like, comment, share, subscribe, and I'll see you in the next video.